Hello friends, welcome to Sam Biology class. Today we shall discuss about direct and indirect ELISA. Direct ELISA is the most simple format. It requires very less time. In this technique, a single antigen specific enzyme conjugated antibody is used for detection. The technique of direct ELISA has four main steps. They are coating, blocking, detection followed by a final read. First step is coating. Coating is the immobilization of antigens on the surface of the microvales of a polystyrene plate. This is achieved by adding antigens to the microvales followed by an incubation for 1 hour at 37 degrees centigrade. After incubation, the plate is washed by phosphate buffer saline in order to remove any potential unbound antibody from the surface. Next step is blocking. Blocking is performed by using agents like bovine serum albumin, ovalbumin, A-proteinin or any other animal protein. This step is necessary in order to cover all the unsaturated surface binding site that will prevent the binding of non-specific antibody to the surface, which will minimize the false positive results. After blocking, the plate is washed by using phosphate buffer saline. Next step is detection. In this step, a single antigen specific enzyme conjugated antibody is added to the reaction mixture so that the enzyme conjugated antibody will bind with the antigen, thereby resulting in the formation of antigen-antibody complex. It is followed by an incubation for 1 hour. The plate is again washed by using phosphate buffer saline, and after that, the next step is final read. In this step, a suitable substrate or chromophore is added to the reaction mixture that results in a color change. When using alkaline phosphatase, the color change is due to the hydrolysis of a phosphate group from the substrate, and in case of horseradish peroxidase, the color change is due to the oxidation of the substrate. The color change is measured by using a suitable instrument such as spectrophotometer. Another format is indirect ELISA, which requires two types of antibodies. One is a primary detection antibody that binds with the antigen, and the other is an enzyme conjugated secondary antibody that binds with the primary detection antibody. So, in indirect ELISA, we need two types of antibodies and there is an additional wash step. Otherwise, remaining steps are identical in direct ELISA and indirect ELISA. This figure explains the process of indirect ELISA. In the first step, the antigens are immobilized on the surface of the microvales of a polystyrene plate. This is achieved by adding antigens to the microvales followed by incubation for 1 hour at 37 degrees centigrade. After that, the plate is washed by a phosphate buffer saline. Next step is blocking. Blocking is achieved by adding suitable agents like bovine serum albumin, ovalbumin, A-proteinin or any other protein followed by incubation for 1 hour. After that, the plate is washed and the next step is the addition of primary detection antibody. After the addition of primary detection antibody, it will bind with the antigen to form the antigen-antibody complex. After 1 hour of incubation, the plate is again washed, and the next step is the addition of enzyme conjugated secondary antibody. The enzyme conjugated secondary antibody will bind to the primary detection antibody. After that, the plate will be washed, and the next step is the addition of a suitable substrate that will result in a color change which can be measured by using specific instrument. Advantages and disadvantages of direct and indirect ELISA. Direct ELISA is quick. It requires only a single type of antigen-specific enzyme conjugated antibody for detection. In direct ELISA, cross-reactivity of secondary antibody is eliminated because in this technique only a single type of antibody is used for detection, whereas in case of indirect ELISA, an additional incubation step is needed. And also there is chances of cross-reactivity with the secondary antibody. But in case of indirect ELISA, a wide variety of labeled secondary antibody are available commercially for detection. Indirect ELISA is versatile because many primary antibodies can be made in one species, and the same labeled secondary antibody can be used for detection. Other advantages of indirect ELISA are maximum immunoreactivity of primary antibody is retained, but in case of direct ELISA, immunoreactivity of primary antibody might be adversely affected by labeling with reporter enzyme. In case of indirect ELISA, the sensitivity is increased because the primary antibody has several epitopes that bind with secondary antibodies. In case of direct ELISA, there is no flexibility in choice of primary antibody level from one experiment to another experiment. But in case of indirect ELISA, different detection methods can be used with the same primary antibody, and in case of direct ELISA, the signal amplification is minimum, whereas in this technique, the signal amplification can be achieved. So friends, this is about advantages and disadvantages of direct and indirect ELISA. For more updates, please subscribe Sam Biology class, and also please share your comments. Thank you.